Hello, everyone, and welcome to the webinar on breeding multi-use naked barley for organic systems by Bridget Mines of Oregon State University. I'm your host, Alice Formiga of eOrganic. eOrganic has many articles, videos, and webinars about organic farming and research, and you can find all of them by typing webinars by eOrganic into a search engine, and you can also find the recordings on the eOrganic YouTube channel. We are recording this webinar, and we will have the recording available on YouTube within 1 to 2 weeks. The webinar will last about 45 minutes to an hour, and then when it's over, we'll have 30 minutes for questions. So I would like to welcome our presenter today, Bridget Mines. Bridget is a postdoctoral research associate with the Barley breeding program at Oregon State University, and she breeds multi-use naked barley for organic systems. She earned her master's from Oregon State University in crop science with a focus in plant breeding and genetics, and her PhD in crop science at Washington State University. So Bridget, I am going to hand over the remote control to you, and as a reminder, you'll want to just click on that once to activate it.
Barley is one of the oldest known domesticated crops in the world, and although it was originally cultivated for human consumption, other end uses have gained importance over time. Barley is the fourth most widely grown cereal in the world, and is the second most widely grown organic small grain in the US. In 2018, approximately 2 million acres of barley was grown in the US. However, in comparison, only about 51,000 acres of that was under certified organic conditions. Barley is a versatile crop with 3 principal end uses: feed and forage, food, and malt. The breeding and production for these end uses has been stratified based on hull adherence and beta-glucan content. So most of the barley grown in the United States has an adhering hull, or what we'll call covered, and a small percentage of the barley that is grown is hullless, or what we'll call naked. However, the exact acreage is unknown because naked barley is currently such a minor crop. So in most barley germplasm, the hull adheres to the grain. However, in some barley germplasm, the hull does not adhere and the grain threshes clean as with wheat. These types are referred to as naked. Naked barley arose by spontaneous mutation about 8,500 years ago. No wild forms of naked barley have been discovered, which indicates that the mutation likely occurred in a domesticated covered type. The adherence of the lemma and palea, or the hull, is determined by alleles at a single locus called nude on chromosome 7HL. When the uppercase nude allele is present, the lemma or palea stick tightly to the caryopsis and remain so post-harvest, as in this image. However, when the lowercase nude allele is present, the lemma and palea thresh freely, as with wheat, and the grain is considered naked. The adhering hull accounts for approximately 10 to 13% of the weight of the harvested barley, and the hull is made up primarily of cellulose, lignin, and silica. Therefore, as a result of the lowercase nude allele, naked barley generally contains greater levels of protein, starch, and total insoluble beta-glucan than covered barley as a result of the dilution effect of the fibrous hull on these components. Here you can see that the naked lines have greater values than the covered lines. However, when we look at the fiber, lignin, arabinoxylans, and cellulose, we see that the covered lines contain more of these than the naked lines. This, of course, is a result of the hull being made up primarily of cellulose and lignin. When we look at physical aspects of the grain, we see that for the covered grain, there's a reduction in kernel weight and number of plump kernels in the naked barley as a result of the loss of the hull, and an increase in test weight and thin kernels, again as a result of the loss of the hull. So we want to talk about some traits of interest that are related to this naked phenotype. While the nude locus, the lowercase nude locus, is responsible for the naked phenotype, the gene affects other traits including thresh ability, resistance to embryo damage or germination, seedling vigor, emergence and establishment, yield, disease resistance, and beta-glucan content. So people have thought that perhaps this is related to the fact that there has been limited breeding for naked barley, or they've also wondered if this is a pleiotropic effect of the nude gene. I want to focus in on a couple of these traits. So thresh ability is the ease of hull removal, and it is an important trait to select for in naked barley because grain with undetached hulls mixed in with naked barley can lower the quality for multiple end uses. So because naked barley is not a commodity crop in the United States, we don't have standards for this. However, in Canada, food grade barley can only contain 5% of the grain with undetached hulls, and feed grade barley can only have 15% of the grain with undetached hulls. So I want you to look at these pictures. And so these are both naked barley lines. However, this one on the bottom has much better thresh ability. You can see there's still a few hulls left in there. And I should say that those are naked barleys where the hull has simply not detached from the grain rather than actually hulled kernels. And this top picture here, we can see there are a great deal more kernels that still have that hull adhering. So studies have been conducted that showed that this trait is controlled by 2 genes, and that additionally good thresh ability is associated with rounder grains that have thin hulls. For our breeding purposes, we can score this visually on using a rating scale. So we would rate this as a much better thresher compared to this line. Additionally, another way to quickly measure thresh ability would be to take both a dirty and a clean test weight. If your dirty and clean test weight are similar, your line has better thresh ability. However, if they're quite different, then you probably have poor thresh ability. The next trait that I want to talk about is resistance to embryo damage. So damage to the embryo can result in poor or uneven germination, which can be an issue especially for seed production and malting. So this is hypothesized to be due in part to the size and shape of the grain. Resistance to embryo damage during grain handling is a trait that is especially important to breed for in naked barley. And we can see here, these lines at the top, their embryo has been completely removed. And while they may still be useful for food or feed end uses, unless sprouting is involved, they're not going to be effective for germination either in the malting process or for seed production. These embryos have been partially damaged. They may still germinate, however, there will most likely be reduced vigor. This is what we're looking for here at the bottom, which is intact embryos. So in addition to breeding, we can also use cultural practices during harvesting and cleaning to reduce embryo damage. And this can include looking at the effect of the harvester drum speed and the distance between the threshing drum and concave, and adjusting these combine settings in order to be a bit more gentle on the grain as it's being harvested. Again, like thresh ability, we can use visual scoring to rate embryo damage. This includes looking at grains like in these 3 pictures and conducting germination tests. So yield is another very important trait that is related to naked versus covered barley. So naked barley has been reported to yield less than covered barley, and researchers hypothesized that the reduced yields of naked barleys could be a negative result of the new gene on yield. However, poor emergence, germination, or reduced seedling vigor can also have an effect on yield. With the weight of the hull being about 10 to 13%, we would naturally expect that much lower yields due to the lack of the hull. Other studies have shown that with adjustments for germination, researchers found that when naked and covered barley lines were planted at similar seeding rates, naked barley had a final plant stand of only about 75% of the covered barley, which was hypothesized to be a result of poor emergence or reduced vigor in the naked lines. Therefore, it's recommended to increase the seeding rate for naked barley compared to covered barley. Another hypothesis for the lower yield in naked barley is that less breeding has been devoted to naked barley. In the last few decades, researchers have begun targeting the naked gene. However, improvements can still be made. And there are other studies that show that naked barley breeding lines can significantly outyield or show no significant difference from the covered checks when the weight of the hull is taken into account, which suggests that it is possible to breed for improved yield in naked barleys through recombination and selection. And anecdotally, from our own trials, we have seen some breeding lines that, even without taking into account the weight of the hull, can be competitive with the covered breeding lines. Another trait that I want to talk about is beta-glucan and how it relates to different end uses. So beta-glucan is a soluble dietary fiber found primarily in barley and oats. Beta-glucan content in the grain has important functionality repercussions for all end uses, and is important to consider when breeding multi-use barley. Beta-glucan found in barley is comprised of beta-1,4,1,3 mixed linkages, and these beta-glucans are easily soluble in water, which results in highly viscous solutions. In the kernel, beta-glucan is primarily found in the cell walls throughout the endosperm, but is also found in the subalurone layers adjacent to the endosperm. Reported naturally occurring levels of beta-glucan content in barley range from about 2 to 11%, but most varieties contain between 4 and 7%. So levels of beta-glucan in the barley are influenced by both the genetics of the grain as well as the environment. Grain beta-glucan is controlled by both quantitative and qualitative genetic components, including a positive pleiotropic effect of the recessive allele at the waxy locus with increased beta-glucan content. So alleles at the waxy locus are encoded by the granule-bound starch synthase 1 gene, and when there's a deletion at this locus, we see increased levels of amylopectin and reduced levels of amylose. So normal starch in barley contains about 25% amylose and 75% amylopectin. However, waxy starch has about 0 to 10% amylose. Breeders can target high beta-glucan by targeting the recessive allele at the waxy locus. However, as I mentioned earlier, both beta-glucan and waxy starch have an impact on functionality and quality for the various end uses. Another trait that we consider when breeding for naked barley is seed coat color. So hulled barleys can also have different seed coat colors. However, they're going to be less visible due to the covering of the hull. So in addition to the normal golden blonde color of barley, barley can have blue, black, or purple seed coats. And so this is due to anthocyanins in the blue and purple seed coat, and the black contains a melanin-like pigment. So the purple and black pigments are located in the pericarp or outer layers of the grain, and the blue is located in the allurone layer. Because they're located in different layers, it's possible for breeders to stack these colors for improved nutritional content. When breeding for these color traits, we need to use land races from areas around the world where barley remains a staple crop, because these lines contain black, blue, and purple seed coats. And breeding for seed coat color can be beneficial for multiple end uses, both because the anthocyanins and melanin-like pigment contribute to total antioxidant capacity of the grain, as well as for the potential market appeal of the attractive colors. So I want to switch focus now a little bit away from traits relating to barley end use and talk a little bit about breeding for organic systems. So most barley varieties are bred for and under conventional systems and may not perform as well under organic conditions. Research has shown that selections that are made under organic conditions are generally suited better for organic production systems. So for our research, all of our trials and end use assessments are conducted under organic conditions. And for the breeding purposes, we're especially focused on targeting for genetic disease resistance, weed competition, and input use efficiency. So in the Pacific Northwest and Western Oregon, we are lucky to have climate conditions that give us a great deal of natural inoculation from fungal pathogens. So we're interested in breeding for genetic resistance to a number of foliar pathogens, stripe, leaf, stem rust, scald, and powdery mildew are all pathogens that we look at and select for resistance to. And organic growers are going to see the most benefit by growing varieties that have genetic resistance. Covered and loose smut are especially concerning for organic systems because these are seed and soil-borne pathogens that can infect one crop one year and then cause problems in the crop the next year. Unfortunately, because of conventional seed fungicide treatments, most breeders have stopped focusing on breeding for smut resistance in barley. There are a few programs that have continued to breed for this, and this is one thing that we'll be looking at. However, organic growers are recommended to look for certified seed or try and find the varieties that have been bred for smut resistance. Additionally, there has been some research that has shown that hot water or steam treatments can be effective in killing the pathogen without killing the seed. However, these have mostly been shown to work on small seed lots. And as far as I'm aware, there aren't large facilities that can currently use these treatments on barley. Ergot is another pathogen that can be a problem, especially in terms of the resulting ergot sclerotia that can cause health issues. So for that pathogen, it's best to simply use good cleaning methods, gravity or sizing equipment. Barley yellow dwarf virus is a virus that's transmitted from aphids and affects cereals as well as wild grasses. In order to reduce this pathogen, using a late planting for fall barley or reducing volunteers and green bridges can help with control of barley yellow dwarf virus. So weed competitive ability is one of the things that we're especially focusing on in our organic breeding research. So barley does have the ability to compete well with weeds. However, there are a few things that will make certain varieties better able to compete. So for our research, we have been conducting stand counts as well as early vigor ratings and early plant height soon after planting. So you can see in this picture, each of these are different varieties, and some do appear to be much more vigorous than others and will most likely do better at outcompeting the early weeds. Later in the season, we have been measuring weed counts in plots and looking at growth habit and canopy coverage as the barley's ability to compete with the weeds. For winter hardiness, barley can be planted in the fall as a winter barley or in the spring. And winter barley does require a period of vernalization, and also winter barleys have a certain level of winter hardiness to allow them to survive through the winter. Spring barleys do not require vernalization and also do not have winter hardiness, so it's recommended to plant them in the spring. Facultative lines do not require vernalization, but generally don't, and generally also have winter hardiness, so they can be planted either in the spring or in the winter. So for our trials, we score for winter survival and frost damage, and in Western Oregon, we very rarely get cold enough temperatures in order to see a lot of winter kill. So we participate in collaborative nurseries, as with this research project, in order to send our lines to colder places so that we can observe differential winter survival. Now I want to transition to talking about different end uses. Most feed barleys have adhering hulls as a consequence of the fact that breeding efforts for the past 100 years have focused largely on breeding for malt barley. When a variety did not meet malt specs, it was used for feed. Approximately 75% of the world barley acres are grown for animal feed. The hull consists of insoluble dietary fiber, which is a benefit for ruminants, but does not have any benefit for non-ruminants or poultry. Seeing the benefit of breeding for naked barley for monogastric feed, breeders in Canada began breeding naked feed barley in the early 1970s. As a result of these efforts, by the late 90s and early 2000s, approximately 750,000 acres of approximately 24 naked barley varieties were being grown in Western Canada. And the success of these varieties, including their rapid acceptance and increase in production, is a result of the collaboration of breeders and animal nutritionists from the initial breeding stages and the subsequent credible reports on nutritional value that were made widely available to growers. So feed barley should provide digestible energy and/or metabolizable energy, which are derived from the starch and sugars of the grain for the growth of the animal. Breeders can focus on selecting for higher levels of digestible energy, high starch levels, and low levels of non-starch polysaccharides, including beta-glucans and arabinoxyleins, to improve feed quality. The primary benefits of using naked barley as a feed is that it contains higher levels of protein than covered barley or corn. Additionally, covered barley has a lower digestible energy, up to about 15% less than naked barley, because of the additional crude fiber content in the hull that either acts as a diluent or physically or chemically inhibits nutrient digestion and absorption by the animal. For swine fed a diet of covered barley, supplementation for metabolizable energy, protein, and lysine from external sources is necessary. However, when fed a naked barley diet, this is not necessary, and naked barley is shown to be a superior feed to covered barley for swine. Naked barley can also be a good feed for poultry. However, barley with high levels of beta-glucan and other non-starch polysaccharides presents concern for poultry. Because of the beta-1,4,1,3 mixed linkages found in barley, they tend to be more highly water soluble, and in young chicks, this can increase the viscosity of intestinal fluids, which leads to a condition known as sticky droppings, which is very undesirable. However, these effects are seen primarily in young chicks, and older hens and roosters are better able to handle the higher viscosity of the unsupplemented naked barley. And although naked barley has gained popularity primarily as swine and poultry feed, research shows that it can also be a good source of energy for cattle. In studies comparing naked and covered barleys and corn, researchers found that at 48% of the diet on a dry matter basis, the naked barley was equal to or better than corn as an energy source, and resulted in greater energy yields and improved overall digestibility compared to the covered barley and corn. So as part of our project, we're collaborating with Dr. Jim Hermes, who's a poultry extension specialist here at OSU, to conduct feeding studies for both layer hens and broilers. These will include several different diets with organic naked barley and wheat or soy, looking at egg quality, bird body weight, and egg number, and body weight in the broiler trial. So stay tuned for results from that study. So now transitioning to our next end use of malting barley. The principal reason for the continued production of barley in most of the world is malting, with most of the malt being destined for brewing and distilling. Barley is the optimum substrate for malting and brewing because it provides a balance of starch and enzymes for the subsequent nutrition of yeast during fermentation. With only a few exceptions, malt barley research has been directed at barley with hulls. In traditional brewing practice, the hulls play a very important role in the brewing step of lautering, where they provide filtration. Additionally, they protect the growing acrospire during the malting process. Advances in brewing technology, including the use of mash filters, as well as directed breeding for malt quality, make it possible to achieve higher brewing efficiencies with naked barley. Naked barley can present an opportunity to the malting and brewing community through significantly higher levels of malt extract and improved beer quality due to the absence of unwanted compounds in the hull, such as tannins and other polyphenols. However, there are some concerns with malting naked barley. These include higher screening losses due to the smaller size of the kernel. Higher temperatures during kilning can result in harder grain and lower friability values due to case hardening. And high beta-glucan content is a problem. Beta-glucans in barley can cause a number of issues for the malting and brewing industry. Malt extract levels are negatively correlated with beta-glucan levels, and high levels of malt extract are desirable. Filtration issues can arise as a result of the viscous extracts produced by the beta-glucans, and undegraded beta-glucans can result in excessive haze or precipitate in the beer. In order to help prevent this, breeders can select for softer kernels, which refers to the kernel hardness, which can improve water absorption and even modification, larger, rounder kernels in order to prevent screening losses and again improve the evenness of modification, and lower to moderate beta-glucan levels in order to reduce issues associated with that. So modification in protein levels can also be problematic, and this may be a result more to the malting process than the barley itself. Modified steep and germination schedules may be necessary for successful malting of naked barley. Researchers have found that naked barley malted with a multi-step steep had higher steep out moistures when combined with longer germination times. The malts also had higher levels of friability, lower beta-glucan levels, and higher Kolbach indices, which all indicate a better modified malt than naked barley when it was malted with traditional malting, steep, and germination schedule. So for our project, using these micro and mini malters that we have here at OSU, we will be making pilot malts with some of the lines from our breeding trials. We're interested in looking at colored barley for malting and brewing, and using this micromalter CLP unit to determine appropriate steep and germination regimes for the different barleys that we're growing in our trials. So moving on to brewing, despite the extensive research on malting naked barley, there are limited data on brewing trials. There are many theoretical advantages to brewing with naked barley, including economic benefits that are associated with reduced freight and storage costs, higher extract levels, and the potential quality advantages, including improved physical stability in the final beer. Researchers determined that when using mash filter technology, well-modified malt had adequate mash filter performance and presented potential advantages in commercial scale brewing. Research using between 50 to 100% naked barley malt bill resulted in successful brews. However, the researchers found that although the extraction efficiencies were lower than expected, the wort gravities were higher than with covered malt. Although these data are limited, what we do see indicates that there is potential for brewing with naked barley, but the brewhouse may need to make some production changes to get the greatest extract efficiency out of the naked malts. For our project, we'll be collaborating with the OSU Fermentation Science program to malt 2 naked lines and a covered check in our mini malter, and then these will be brewed using both a lauter tun and a mash filter in order to compare brewing efficiencies. The distilling community has also expressed some interest in naked barley for whiskey as a means of increasing alcohol yield. Distilleries use mash tuns to filter the wort, and the hull particles are necessary for creating a filter bed. However, recent research has shown that the extent of malt modification may be more important for rapid filtration than the hull. Researchers conducted studies to determine the potential of naked barley for the Scotch whiskey industry. Using 2 naked barleys and a modified steep schedule, they found that the naked barley malt had good levels of amylolytic enzymes, including dextrinizing units and diastatic power. These naked malts produced wort that had shorter filter times and had much higher predicted spirit yield than the covered check. These results indicate that naked barley could benefit distillers by providing higher alcohol yields as well as increased throughput. Food barley has a rich and traditional culinary significance in many areas around the world. However, in the United States, it has almost entirely disappeared as a food. Recently, barley foods are on the rebound, especially with consumers who are more likely to have a heightened awareness of fiber and whole grain nutrition. Barley has the advantage of providing fiber and other healthful components in a package that has half the fat content or less compared to oats, and greater total dietary fiber content than wheat, oat, or rye. Numerous clinical studies have been conducted on the positive effects of beta-glucan on human health, focusing on the benefits of beta-glucans for lowering postprandial blood glucose levels, the lowering of plasma LDL cholesterol concentrations, and as a protection against mutagenic agents. The capacity of barley foods to reduce cholesterol was a key factor in approval of the FDA health claim for barley in 2006, and similar claims in Europe in 2011 and Canada in 2012. Food barley can be used in multiple applications and can be ground into flour, cracked into grits, flaked, steamed, boiled, toasted, extruded, and roasted. For these it can be made into baked products, including risen and flatbreads, although not at 100% due to the lower gluten content, pastries, pancakes and cookies, noodles and pasta, tortillas, tea and other flavored beverages, and porridges and other breakfast cereals. In order for varieties to be successful and well received by producers, bakers, and chefs, breeders can collaborate with cereal chemists, including Dr. Andrew Ross here at OSU, to characterize naked barley lines for cereal quality traits prior to the development of food products. The resulting information can be critical to breeders selecting for food quality in their programs. We will be collaborating with Dr. Ross to measure food quality functionality traits, as well as do some product development work on the naked barley lines in our trials. So for breeders, there are a number of different traits that we can target when breeding for food barley, including beta-glucan content, protein, kernel hardness, whole grain nutrition, as compared to pearled barley, which is hulled barley that has had the hull as well as some of the bran and germ removed and is no longer considered a whole grain. And then the functionality related to these other traits, minerals and antioxidant capacity. So as with the other end uses, beta-glucans, as well as starch type, are one of the most important factors in influencing functionality of food products, including flour yield. So genotypes that have high levels of beta-glucan resist breakdown and produce a greater proportion of larger sized particles during roller milling. In comparison, low beta-glucan varieties have thinner cell walls and are easier to mill. Dough properties are also altered by the levels of beta-glucan in the grain. We see dramatic increase in water absorption with high beta-glucan barleys, increased dough strength, reduced dough resistance to extension, and impacts on starch pasting properties, lowered gel viscosity, and increased gelatinization temperature. Although breeders have only been breeding for food barley in the United States for the last few decades, there are a number of functionality traits that are important to look at. So for our project, we will be measuring a number of these physical and chemical traits, as well as adding to this list in order to determine which quality traits are important for selecting for food barley. From both our malt and food research, we've started doing some sensory evaluation, including hedonic, preference, and descriptive tastings at various events using steamed grain, bread, crackers, digestives, biscuits, roasted tea, and hot steep. On these ballots, we've been interested in getting people's thoughts on flavor, texture, aroma, color, and overall liking or disliking. This can help us select new varieties and find out people's preferences for naked barley. So I've been so far giving you a bit of background on breeding naked barley for multiple end uses. However, this is all leading up to our current project, which is a grant funded by USDA NIFA OREI in 2017, entitled Developing Multi-Use Naked Barley for Organic Farming Systems. OSU is the lead institution on this grant, and the states that we're participating with are Washington, Minnesota, Wisconsin, and New York. As part of this grant, we'll be conducting research, extension. We have education component as well. We're looking at evaluating agronomic, food, feed, and malt quality traits under organic conditions, and measuring the economic, environmental, and health benefits of organic naked barley production and products. So some of the resources that we have in terms of germplasm include the Oregon Naked Barley Blend, or the ONBB, which is a blend of 753 pure lines that are all naked, but otherwise quite phenotypically different. And so far, through various distribution means, we've given out about 20 kilograms of seed. If you're interested in getting a small amount of seed, my email address is included at the end of this presentation, and we'd be happy to send you some. It's a released germplasm through OSU. You can select your own variety or grow it as a blend. Additionally, we have the diversity panel, which is 384 genetically diverse lines, including land races, breeding lines, and released varieties from programs around the country and around the world. We have 20 advanced naked lines that are being grown in multi-region agronomic yield trials. We also have on-farm production with our most advanced breeding lines or recent releases that are being grown in 1 acre blocks with farmers to evaluate growing potential as well as end-use quality on a larger amount of grain. So for these field trials, as I said, they're being grown in these 5 states around the country, both in the fall and in the spring. On the regional and diversity panel, we're evaluating for agronomic traits, weed competitive ability and resistance to biotic and abiotic stresses, as well as quality traits on the regional trial and the increase blocks. And then the diversity panel will be genotyped and will perform genome-wide association studies to look at genetic control of the traits that we're observing. This project also includes a very large outreach component. So far, we have done and are continuing and will be doing workshops on best management practices, the benefits, processing procedures for naked barley, targeting towards students, growers, gardeners, researchers, processors, and consumers. These outreach events have included Barley Day 2018, which is our annual field day that we hold here in Corvallis. We had about 100 people show up, and this last year's focused on this project. We've been lucky to participate in the Culinary Breeding Network Variety Showcase for a number of years, collaborating with Lane Selman on the sensory exploration components. We've had tastings, workshops, presentations at the Organic Seed Growers Conference and Organicology, the Grain Gathering, Cascadia Grains Conference, as well as additional workshops and classes. We're also very lucky to be collaborating with Green Market Grains, which is part of Grow NYC, in conducting other outreach opportunities. And then for our education component, we're working with K-12 schools, and each state has a lead teacher that we've identified, and we're working with them to collaborate on developing lesson plans around naked barley using the Oregon Naked Barley Blend as a learning tool. And these collaborations have included field trips where the kids have come out to the greenhouses or farms to learn about what we're doing out there, as well as us visiting the classrooms, which has included workshops as well as eating pancakes. And through these collaborations, we are hoping to teach the kids a little bit about math, science, agriculture, and nutrition related to naked barley. And so far, we've seen good results from this. The kids seem to really like it. So finally, after all of that, in conclusion, breeding multi-use naked barley can give organic farmers a second or third option for sales. Naked barley, when bred for multi-uses, should have moderate levels of beta-glucan and normal starch. And farmers, processors, producers, and consumers can benefit from naked barley. However, education is going to be necessary for acceptance. And then finally, I'd like to acknowledge all the collaborators on the grant who've been so great to work with, as well as the wonderful Barley World crew here in Corvallis. This is our grant number, if anyone's interested. We have several web resources, including a website on the eOrganic website, as well as a page on our barleyworld.org, which is the OSU Barley Breeding Program website. We do have Instagram and Facebook accounts at MultiBarley. You can go check those out and follow along with what we're doing. And then I also want to mention that for our grant, we'll be giving 3 more webinars related to this project. and we'd love to do them on things that are especially interesting to people. So if you have any suggestions on what you'd like to hear more about, most likely they'll be focused on the different end uses. You can shoot me an email. You can also ask me any questions, follow up, or request some of the ONBB to grow on your own. So with that, I think Alice is going to open it up to questions.
Yeah, thank you, Bridget. We're going to start our question and answer session. We have about a half an hour available for that. So I'd like to I'd like to encourage anybody who has a question, because very likely you will have the opportunity to get it answered by Bridget. So you can just type it into the Q&A box on your screen, and we'll be reading the questions out loud until either we run out of time or we run out of questions. So if you don't see the Q&A box there should be a black bar with some controls at the bottom of your screen, and if you click on the Q&A, that should pull it up. I just wanted to let everybody know, in case you came on a little late, that this presentation is being recorded, and you'll be able to find it on the eOrganic YouTube channel in about a week. And I also put a link to a handout of the slides in the chat box, and that's going to be available on the e-extension page, which I'll put up in a second, with the archived webinar embedded there as well. So hopefully now we'll have a few questions. We had a comment during the webinar by Steve here. He says, interesting work going on here in Skagit County with Foothills Farm pasture poultry, and making a 2-day fermented barley wheat liquid mash as a feed source. And they're studying it now for better uptake and conversion factors. Okay.
Great.
Yeah, that does sound interesting. So if anybody else has a question, we don't actually have a lot of questions, so please feel free to take this opportunity. Okay, so here we go. Are farmers involved in assessment and scoring of on-farm barley trials?
So right now, our on-farm trials have mostly consisted of the larger 1-acre increase block, and that has been done with released varieties up until now. We will be doing an advanced breeding line. However, we would love to have farmers come and look at our plots on station, and I mean, this is a great opportunity for participatory plant breeding, and right now, you know, is a great time to do that. So, in my experience, trying to do participatory breeding with grain farmers, they have to be really excited about it. Mostly, they come to the plots and seem happy, but don't have necessarily a lot of opinions. But there aren't a lot of farmers currently growing naked barley. So we would love for them to come out and look at the trials and let us know what they think.
Okay. What considerations have you made for dwarf stature in regards to lodging issues?
So we do measure plant height on all of our breeding lines, and lodging is certainly an issue for quality as well as yield. So we are breeding for shorter barleys, but not necessarily like dwarf varieties. We also tend to see in our organic plots lines that may lodge in the conventional nurseries that have higher nitrogen inputs tend to be a little shorter in the organic conditions and don't lodge as much. But that is something that we're looking for.
Okay. How many genes control the different kernel colors?
Oh, that's a good question. So currently what is known is that the black trait is controlled by a single gene with an allelic series. So there are different levels of black that can occur. Purple is controlled by 2 genes, and blue is actually controlled by 5 additive genes. So we can use like marker-assisted selection to breed for higher levels of color.
Do you have a specific ideal barley that you're selecting for right now? And if so, how far are you from some kind of finished variety?
That's a good question. We have a couple of naked releases, naked barley releases, I should say, from the OSU program, including Streaker, which is actually this picture here at the bottom is a blend of 3 sister lines, and it's a 6-row winter type. We also have recently released Buck, which is also a 6-row winter type, but it's a pure line. Both of them have some issues with disease, and so really what I would love to see is a disease-resistant barley that I'm particularly like the color, especially for the naked lines, because I think that may have some appeal to consumers who are seeing barley as sort of a new crop, something different than maybe the pearl barley in their grandma's beef and barley soup. And so right now, one of the things that I'm focusing on in my crossing block this winter is a winter purple 2-row. And we do have one that is pretty close, but it is a bit tall and does have some lodging issues, so I'd like to work on shortening it a little bit. And in terms of how far out we are, if we do decide to release the purple one, it could be in the next year or 2. If we're starting from scratch, it could be more like 10 years.
Okay. What do you think is the best yield comparator for naked barley, covered barley or wheat?
Did Pat ask that question?
Yes.
Yeah, thanks, Pat. Pat is actually the PI for this grant. In terms of test weight, I forgot to mention this earlier, we do use wheat as a comparison rather than the typical hulled barley test weight of standard of 48 pounds per bushel. We use wheat test weight at more like 60 pounds per bushel. I think for multi-use naked barley, both of them are probably good comparators, because for malt barley, you're going to be comparing to covered barley, and for food, you're probably going to be comparing to wheat. So really, ideally, we should be just trying to out-yield both wheat and covered barley.
Okay, David says, we have David from Timeless Seeds in Ulm, Montana. We have had both beer and whiskey brewed and distilled by a local brewery and a local distillery, and they also grow and package a purple naked barley. So he is very much looking forward to following your work and collaborating when possible. So thank you, David.
Yeah,
We'll
I
connect you up.
have a package of your purple naked barley.
Oh, good.
And
Okay.
it's quite tasty, and I would love to talk more.
Okay, let's see. Can you expand more on the environmental benefits of barley, specifically the ecosystem services?
Yeah, so I guess in comparison to, say, wheat, barley requires less nitrogen inputs and has a shorter growing period, so we can get more rotational benefits, which could be considered an environmental benefit. You know, in organic systems, the barley can help, or the barley straw that's tilled back into the soil can provide organic matter and break disease cycles for the following crops. We do have someone on our team who will be looking at both the economic and environmental benefits in order to sort of quantify them a little more after this project is over.
Okay, great. Let's see. Here's some of since the naked barley seems like it has a long way to go. Will it be worth the effort?
That's my mom. Thanks, Mom. So I guess I would perhaps disagree that it has a long way to go. I think that we're already seeing people getting really excited about this, and breeding just in general is a long process. So we have identified lines both from our advanced trials and the diversity panel that do well in organic systems. And I think that really the issue is consumer acceptance, processor acceptance, willingness to work with what is essentially a new crop. And I think that giving farmers another end use option for sales really could be worth the benefit.
Okay. Let's see, here's David again. In our observation, growing a purple naked barley for nearly 2 decades, both low fertility and drought stress can have a negative impact on the purple color. Is there research that explores stress on coloration?
That's a good question. So we do definitely see color differences as a result of the environment. And it would be, going back actually to my dissertation work, I did quite a bit of work on color and the total antioxidant capacity of colored grains. And we did have a few different locations that had different levels of stress, and we did see slightly different colors. However, those have yet to be quantified. And there isn't really any formal research that's being done looking at stress on color, but that's something that would be worth looking into. I will say another sort of interesting note about the purple lines in particular is that we do see that the anthocyanin levels peak several weeks before harvest. And actually, when we harvested the grain at a higher moisture and then dried it down, those kernels were definitely more purple than kernels that were harvested at the appropriate moisture. So I've definitely had interest in doing further research on color barley, and perhaps now I should incorporate some stressed environments to look at the impact of that on coloration. So thanks.
Yeah. If someone planted your Oregon Naked Barley blend in Southeast Michigan and selected for a particular trait, how many generations of planting and regrowing would it take before the new line appears unique? In other words, is barley fast to respond to environmental and selection pressures?
Let's see. Where is that question? I want to.
It's in the chat.
Okay. Yeah, so I'll tell you a little bit more about the Oregon Naked Barley blend, is that it is a blend of 750 doubled haploids. So those are all pure lines. So if you were to select a single head out of that population and just plant that head, you would have a pure line the next year. In terms of looking at natural, if you were to grow the blend and look at natural versus artificial or human selection, I guess it would sort of depend on how strong that pressure is before you would see a response in the population. So for instance, if you were selecting against stripe rust, we see varying levels of stripe rust in our lines. Some so severe that there's no seed production, or the seeds are sterile. And so if it were so severe, you would see fairly quickly. However, if it's just a foliar problem that sort of is problematic each year, but does not actually kill the grain, it would probably take quite a bit longer. And then it looks like the next part of that question, how do you describe the difference in taste of different colored barley?
Yep.
Ooh. Well, taste is very subjective. And so, you know, everyone's going to taste something a little bit different, but we have heard words such as like nuttier being used to apply to the darker varieties.
Okay, let's see. We have several questions that are interested in the education component, so I'll just read them one at a time here.
Okay.
Let's see. With younger students, I am trying to do something similar, especially around STEM. Can you elaborate a bit on the classroom work, age levels, activities, and support materials or contacts in Washington State? My point of entry is the beginning of agriculture, like ancient Mesopotamia, and the role barley played in the invention of agriculture, plus cuneiform writing system, math, accounting, numbers, and many other aspects. He'd be happy to discuss this further offline, which sounds like it might be a very good idea, but if generally you can elaborate a little bit more about that.
Mm-hmm. Yeah, I know Steve, so we can talk later, but just briefly, here in Oregon, we've worked with middle schoolers, so 6th to 8th grade, and that has included, we had last year, we did, we gave them the Oregon Naked Barley Blend, which also includes different colored grain, and they selected out the different colors to make a histogram, and then we hypothesized that when we planted a blue kernel, the next year from that plant we would get a blue kernel. So that may be a little bit more advanced than what you're looking for with younger students, but we're also doing some ecosystem or natural versus agro ecosystem work this year, looking at barley versus native grasses and their response to nitrogen. So we have a few different lesson plans that we've come up with, and I believe that we're willing to share those. And I think that we also do have a bulletin that we've posted at the eOrganic website, but I can check back, and if it's not there, we can get that posted.
Yeah, the link to that website is right here on the screen under web. We have some other interesting resources from Bridget's project, for example, some recipes, including how to roast barley for tea. So you might want to check out both of these websites here. Let's see, another education question. I'm very interested in the school's involvement. Are you getting the school children engaged in feeding back useful information? And is this also being looked on as a way of increasing early interest in the plant and food sciences?
So I'm guessing that would be like asking the students for their feedback. We haven't done that in an official capacity because doing research on school children does require some institutional review board approval. However, I would say, so last year we had, I think, about 150 middle schoolers come out to our research plots, and we did get a lot of informal feedback from them. We had different stations on soils, diseases, food and nutrition, sort of planting, and there's one more, breeding and crossing, where they actually got a chance to do some emasculations and pollinations. And then they were all asked to fill out assignments and answer questions. And I was at the food station where we were giving them little treats. So, they did certainly enjoy that. But I think overall, we got positive feedback, but not necessarily for like research purposes. And I think, especially that field trip setting, the kids really, some of the kids, I mean, these are middle schoolers, really like took some of that away with them, or at least we hope that they did. But just, informally talking to them after, a bunch of them really seemed to get a lot out of it.
Yeah, I guess he said he heard of a Saturday class for 14-year-olds involved in genetics of soy in Eastern Europe a while back, and many of them grew up to be plant breeders.
Yeah, I mean, who knows what impact we have on these kids, but it would be great. It's great to introduce them to plant breeding and agriculture as young as possible.
Yeah, let's see. Can you elaborate about the methods and results of your sensory evaluations? And I just wanted to mention that we just published an article on eOrganic, and while you're talking about the question, I will find that article and type a link to it in the chat box in case the person would be interested in reading it, because I believe you saw that article as well. So.
Yes, and in fact, that's a great resource, because what we're doing is in collaboration with Lane Selman at the Culinary Breeding Network, and she's one of the authors on that article. And although it is directed at vegetables, a lot of it applies to grain. So what we have been doing so far is what Lane likes to describe as sensory exploration, because it is extremely informal. We are looking to conduct more formal sensory evaluations in collaboration with the OSU Food Science and Sensory Lab. However, the ballots that Alice shared in this article are quite similar to what we use. Generally, we do 3 to 4 different varieties of barley in anywhere between 1 to 4 applications. So I think the most complex one that I have done was 4 varieties in 4 applications. So that was, we did bread, steamed grain, roasted barley tea, and little cracker biscuit things. And then we did both a descriptive sensory on the bread as well as hedonic sensory on the other 3 applications, in which people had to rate from 1 to 9 how much they liked each of these, the grain, the steamed grain, crackers, and roasted barley tea for I think flavor, texture, appearance, and overall. And then for the descriptive tasting that we did with the bread, we actually provided them with a list of descriptors, and they were asked to, I used that list as well as their own words to describe the 4 different breads. And we've done these sensory explorations, both that particular one happened at the Grain Gathering Conference in the Skagit Valley of Washington, and in that case the participants were excellent tasters. It was a lot of bakers and people involved in the grains movement, and so they gave us a lot of great words and responses. We also do these tastings at events like Organicology and the Organic Seed Growers Conference, where people are just sort of passing through tasting things, and so they may not take it so seriously. So we've had some that have given us more quality information, but so far we haven't published anything, and we're just sort of internalizing these data. But again, take a look at that article that Alice just posted, because It's a great resource on how to conduct informal sensory evaluations, and
Yeah,
the
it's
methods that they
called
use are similar to what we do. Yeah.
yeah, it's called Simple Methods for Market Growers to Assess Culinary Quality with Consumers, and it's written by people that have been doing that out of the University of Wisconsin with a group called the Seed to Kitchen Collaborative, and then also the Culinary Breeding Network, which Lane Selman runs here in Oregon. So that link is in the chat box there. Okay, we have one more question here. Is there much work focusing on linking phenotypic characteristics and genomic expression?
So we will be, as I mentioned with the diversity panel, we are currently having that genotyped on the 50K SNP chip. And so we'll be using that to associate phenotypic characteristics with the genotype data that we get. However, for naked barley, there hasn't really been much done on that. However, the naked gene is 1 out of 30,000, and there has been lots of work focusing on linking phenotypic characteristics.
Okay, great. Thanks everyone for all these great questions. And as I mentioned, you'll be able to find the recording in about a week on the eOrganic YouTube channel. And we would also be very grateful to you if you could fill out the follow-up email survey, which we're going to be sending sometime this afternoon. So thank you so much, Bridget. And
thanks, Alice, and everyone for attending.
Yeah, it's great to have you, and we are definitely looking forward to learning more as the project progresses. So I hope everyone can join us for the many other webinars that eOrganic is offering on a huge variety of organic farming and research topics on our schedule this winter. And you can find those simply by typing webinars by eOrganic into a search engine. So thanks again, Bridget, and thanks for everyone for joining us.